Welcome back to the Deep Dive. Today, we have a really ambitious mission. We're going to try and transform the incredibly intricate blueprints of how the ear develops embryologically into a clear mental map for you.
Yeah, we are plunging deep into the source material here, looking at the chronology, the spatial relationships. It's all laid out in these classic anatomical texts.
Okay, let's unpack this because the ear is just a symphony of coordinated effort. We're tracking three completely distinct regions: inner, middle, and external ear.
And they have to develop at the same time, often from totally different embryonic origins, and then connect perfectly. It's a masterclass in biological manufacturing.
It really is. And the challenge for us, is to visualize this whole three-dimensional choreography without diagrams.
Exactly. We're moving from just simple cells to this coiled labyrinth and these tiny, delicate ossicles. The precision is just fascinating.
Absolutely. So let's start at the foundation, the inner ear. This is the labyrinth, and it's responsible for both hearing and our sense of balance.
And it starts incredibly early. We're talking right after the very first signs of eye development.
So where do we begin? What's the first tissue we should be looking at?
It all starts with the ectoderm, the outer layer of the embryo. At stage 9, which is, very early, you see the otic placodes.
Yeah. What are those exactly?
Think of them as just two small, thick patches of the surface ectoderm. They're sitting right next to the hindbrain.
Okay, so you have these two patches. What's the next critical step? Do they just fold inward?
They do, exactly. The placodes start to invaginate, that's the term for folding inward, and they form what's called the otic pit.
A little depression.
A little depression, and by stage 12, so around 30 to 32 days, that pit pinches off completely from the surface, and that separation is key.
And what are you left with?
You're left with a simple hollow sac of epithelium just sort of floating beneath the surface. That is the otic vesicle, or otocyst.
And this tiny little sac is the starting point for the entire complex labyrinth.
The entire thing,
Yeah.
yes.
Here's where it gets really interesting for me. How does that simple hollow sac morph into the utricle, the saccule, and those three semicircular canals? It seems like an impossible transformation.
It starts differentiating really fast at stage 14.
Mm.
The vesicle loses its simple piriform shape. The little tubular structure grows out dorsomedially. That's the endolymphatic appendage.
Okay.
And the rest of it becomes the utriculo-saccular chamber. That appendage then elongates and becomes the endolymphatic duct and sac, which is super important later for managing fluid pressure.
And now for the balance organs, right? The semicircular canals. Those three loops are just vital.
They are. They emerge from the upper part, the pars superior of that chamber. You get two little plate-like diverticula, one growing vertically, one horizontally.
Okay, so two flat plates. How do you get three loops out of two plates?
This is the really cool part. The source material describes this amazing process where the epithelia at the center of these plates actually touch and fuse together. It forms a fusion plate.
So it seals up in the middle first.
Precisely. And then the center of that fusion plate is resorbed. It just dissolves away.
Oh, wow. It's like sculpting by subtraction.
It is. Think of it like taking a sheet of paper, folding it, and then punching a hole in the middle. The edges are left behind as the canals. The vertical plate makes the anterior and posterior canals, and the horizontal one makes the lateral canal.
That's fantastic detail. It really helps you picture it. So while that's happening, what's going on with the main vestibular chamber in the cochlea?
Well, that utriculo-saccular chamber divides. A
Yeah.
horizontal fold appears, and you get the larger dorsal utricle and the smaller ventral saccule.
And I stay connected.
They do, but only through a very narrow little tube, the utriculo-saccular duct. And here's another critical point. The whole labyrinth rotates during this period. Its long axis moves from vertical to horizontal.
So it's reorienting itself as it specializes. Okay, and what about the cochlea, the actual sound receptor?
The hearing part, right. So that starts as an elongation from the ventral tip of the pars inferior. By stage 15, you can see the early duct, the cochlear and login.
And its defining feature is that coiling process.
Yes, the distal part starts to coil, and it keeps going until it hits 2.5 turns, which is usually around postmenstrual week 10.
And within that delicate coil, that's where we find the organ of Corti.
Correct. The central domain of that duct goes through this intense differentiation to become the organ of Corti, which holds the mechanosensory hair cells. And the molecular choreography here is just exquisite. You have signals like BMP and Notch that are defining which cells become sensory and which become support cells.
It's an incredibly sensitive process. So once that membranous labyrinth is formed, it needs protection, right? It needs a bony house.
It does. The surrounding mesenchyme first turns into cartilage, forming the otic capsule. Then, between weeks 16 and 23, that cartilage starts to ossify. It turns to bone, forming the bony labyrinth inside the temporal bone.
What about the fluid spaces around it, the perilymph?
The perilymphatic spaces, the scala vestibuli and scala tympani, they form at the same time.
And how do those channels appear? Are they carved out?
It's more like they form from coalescing vacuoles. Imagine tiny little fluid pockets appearing in the tissue around the duct. They all start linking up, getting bigger, and eventually they connect at the very tip through a little opening called the helicotrema.
Okay, so what does this all mean? We have the structure, now we need the wiring to send signals to the brain.
Exactly. So we end up with 6 specialized sensory patches. You have 3 cristas in the canals for rotational movement, 2 maculae for gravity and linear acceleration, and of course the organ of Corti for sound.
And the nerve connecting all of this, cranial nervate.
The vestibulocochlear ganglion. It appears early, at stage 13. And here's a key nugget of knowledge from the source material. The neuroblasts that form this ganglion come exclusively from the otic vesicle itself.
That's unusual, isn't it?
It's very unusual. Most cranial ganglia have a dual origin from both the placode and the neural crest. The fact that this one is purely placodal just highlights that deep intrinsic connection between the sense organ and its nerve.
We always focus on the sensory input, the signals going to the brain, but sound needs modulation, doesn't it? What about the feedback system?
That's a great point. The source notes the embryological bundle. These are axons that actually start deep in the brainstem, in the pons, and they travel outward to the ear.
So the brain is talking back to the ear.
It is. They primarily connect to the outer hair cells in the organ of Corti. And this suggests the brain is actively modulating our hearing, probably to help improve the signal-to-noise ratio in loud environments.
Fascinating. So that moves us out of the inner ear and into the middle ear. And this is a whole different ball game, right? We're into pharyngeal arch territory now.
Exactly. We shift to structures that come from the pharynx, the pharyngotympanic tube, what most people call the eustachian tube, and the tympanic cavities start as these little outpouchings of the first pharyngeal pouches.
And how does their position change over time?
Well, initially they're sort of inferolateral, so below and to the side of the otic capsule. But as that capsule grows, it stretches them out, widens the cavity, and pulls them into that anterolateral position we recognize.
And the ossicles malleus, inguinus, and stapes, the stars of the show for sound conduction. Where do they come from?
They're all derived from neural crest cells that migrate into those pharyngeal arches. This is so important for understanding why many congenital anomalies affect both the face and the middle ear at the same time.
Okay, so which arch is which?
The malleus and incus come from the dorsal end of the first pharyngeal arch cartilage, and the stapes comes from the second pharyngeal arch.
The stapes has that very distinct stirrup shape with a hole in the middle. How is that formed?
It's formed by a blood vessel, the stapedial artery. The cartilage literally develops in a ring around this artery. Later on, the artery mostly disappears, but it leaves behind that characteristic shape.
And the footplate, the part that actually connects to the inner ear.
That's unique. The footplate forms separately within the otic capsule from a different tissue called paraxial mesenchyme. It's another example of these different embryonic origins all converging perfectly.
And what about the tiny muscles that control them?
The tensor tympani is from the first arch, and the stapedius is from the second arch. It reinforces that whole arch relationship.
And here's a critical detail I found fascinating. The middle ear cavity, it isn't actually hollow right away, is it?
No, not at all. And that's a huge point. It's initially completely filled with this neural crest mesenchyme.
So have to clear out.
It does. That mesenchyme only starts to retract really late, between postmenstrual weeks 29 and 32. This retraction is what suspends the ossicles with ligaments and allows the cavity to expand, but it only fills with air after birth.
With the baby's first cry.
That's right. The first cry opens the pharyngotympanic tube and aerates the space.
Okay, finally, let's move to the external ear, where the sound first enters.
The external acoustic meatus, or the ear canal. This forms from ectodermal tissue from that first pharyngeal arch and cleft region. It starts by folding inward and forming a solid plug of cells called the medial plate.
Wait, it's solid at first. So, like the semicircular canals, it has to become hollow later.
Precisely. The cells inside that solid plug have to separate. They have to canalize. That happens between weeks 13 and 14, and you get an open canal by about week 18. But the bony part of the canal, that develops much, much later, mostly after birth.
The timeline is just staggering. And the tympanic membrane, the eardrum, This must be where all three regions finally meet.
It is the ultimate meeting point. It's a classic trilaminar structure. The outer layer is ectoderm from the medial plate.
Right.
The inner layer is endoderm from the middle ear cavity. And the layer in between is neural crest mesenchyme. And the source makes a great point that in a newborn, the eardrum and the ossicles are already adult-sized.
But the ear canal is still very short.
Exactly.
And the visible part, the auricle.
That largely comes from the second pharyngeal arch mesenchyme. The tragus comes from the first arch, but most of what you see is from the second. And the earlobe, the lobule, that's usually the very last part to finish developing.
This incredible precision means there are so many points where things can go wrong. Let's touch on the clinical relevance, like hereditary deafness.
Yeah, it's the most common congenital anomaly. It affects somewhere between 2 to 6 per 1,000 newborns. You have non-syndromic, which is usually sensorineural, affecting the inner ear.
And syndrome.
And syndromic, which involves other organ systems and can be, conductive or mixed hearing loss. Things like a cochlea with fewer terms or defects in the molecular machinery of the hair cells.
Let's end by focusing on function. When does this whole system actually start working in the womb?
It starts working quite early, actually. Hearing onset is around postmenstrual week 20, but, and this is important, the fetus hears primarily through bone conduction.
Not air conduction.
Right. The sound vibrates through the mother's body and the amniotic fluid. The air conduction mechanism isn't functional yet.
And this early acoustic exposure, it's critical for brain development, isn't it?
Absolutely. The source highlights that if that auditory input is lost, it really negatively impacts the postnatal patterning of the temporal cortex. It's very similar to how the visual cortex is affected by loss of sight.
Which underscores the urgency of intervention if there's a problem.
Precisely. Congenital deafness causes big delays in speech acquisition. So therapies like cochlear implants are optimal if they're implemented within that critical developmental window, which is really the first 1 to 3 years of life.
What a journey. We've gone from that simple otic vesicle to the fully coiled inner labyrinth, the middle ear's reliance on the pharyngeal arches, and then finally the external ear where all three layers meet at the eardrum.
And it really raises an important question to think about. Consider the profound influence of that earliest acoustic input, starting around week 20, and how that initial auditory world in the room immediately starts laying the groundwork for cortical specialization and language processing long before a child ever hears sound through the air.
Thank you so much for joining this deep dive into the anatomical basis of ear development. We really hope this roadmap has given you a clearer picture of these incredibly complex structures and their formation.
And on behalf of the Last Minute Lecture team, thank you for listening.